To wrap up signaling, I thought I would do a couple of sort of synthesizing discussions. The first involves describing what happens with the activated insulin receptor. And the thing about that is that there's really nothing new provided by that. It's just that it uses everything that we've talked about in one way or another. So it's sort of a good summarizing topic. Here's insulin binding to the receptor, leading to autocatalytic phosphorylation of the insulin receptor. That doesn't recruit proteins directly, actually. Well, it recruits one protein directly, but that protein is a new one for us, the so-called IRS protein. And this interaction is one of the few, the only one that we're going to be talking about that involves that other thing besides the SH2, phosphotyrosine binding domain, because that's what IRS1 uses. But in any case, the active insulin receptor recruits and then phosphorylates IRS1. It's a pretty big protein. It has lots of tyrosines on it, and that creates just battery of phosphotyrosines that are recognized by other proteins. So as it turns out, Schick and GRAB2, two adapter proteins, bind, as does PI3 kinase, to other regions of the same protein. So you get this coordinated response involving everything that an activated receptor tyrosine kinase could do. So here's the MAP kinase pathway leading to the things that it does. Here's PI3 kinase activating AKT, and then some of the things that I talked about and some that I didn't. Here is FOXO leading to survival, GSK3 leading to glycogen synthesis and also growth. AS160 is the thing that's responsible for or regulating the GLUT4 from way back when we talked about that process by which insulin causes GLUT4 to migrate from vesicles to the plasma membrane. That's regulated through AKT as a result of the phosphorylation of AS160. And here's mTORC2 complex 1 activating AKT such that it activates mTOR complex 1, and that goes on to, for one thing, inhibit autophagy from a few videos back, and also lead to cell growth through its control of protein synthesis. So that's one way of summarizing everything that we've talked about. And another way that I think might help you once the exam comes around is to think in terms of the receptor tyrosine kinase business as just being the interplay of different binding domains. So you can go a long way just by keeping track of what has which binding domain. So for example, if something has an SH2, then it's going to be the first thing that binds to the receptor tyrosine kinases, and that includes GRAB, PI3 kinase, phospholipase C. We didn't talk about STAT, but it's another one of those things, and of course SARC. What has SH3 is, of course, SARC. The adapter proteins like GRAB2 and Schick. PI3 kinase has SH3, as does these two proteins. There's RAS-GAP, which is essentially activated by the same processes that lead to the activation of RAS. It's just one step behind it, basically. And then that plextrin homology group is found on these proteins and plenty of others. Of course, it's on plextrin, which is that subunit of protein kinase C, and is the reason why protein kinase C migrates to the membrane during the IP3 diacylglycerol pathway from way back when. But there's AKT that we talked about, IRS1 associated with insulin response. SAS is activated through its pH domain as well. Here's a link between receptor tyrosine kinases and the control of G protein coupled responses, because this is the G protein receptor kinase. We didn't talk about Dynamin, so I'll just skip that. So I wanted to leave things with a specific description of crosstalk, because you have a figure in your text, 1555, that covers that fairly nicely. So we talked about all these pathways now, and you can see, how they stem from activated receptors of the different types. But because, for example, receptor tyrosine kinase as well as G proteins can activate phospholipase C, you can get that calcium spike associated with that pathway and all of the things that are associated with the activation of calcium spikes, including calmodulin, protein kinase C, and as we'll talk about with nerves, neurotransmitter release, those things can be controlled by either of those pathways. The individual pathways of the receptor tyrosine kinase crosstalk like crazy, but you have plenty of examples in which the activated components early on in the pathway lead to activation of later components in other pathways. So that by coordinating those controls, you can account for most of the responses that cells make to extracellular signals. And then finally, as another sort of wrap-up diagram, here's a really nice diagram from a really nice Wikipedia page on cell signaling that I highly recommend. So we've essentially covered this part of the signaling pathways, and we're not going to do cytokines at all. So what's left is some signaling that comes from cell-cell coordination, and that's going to be these things over on this side. And then there is the survival versus death signaling that is associated with apoptosis, the last thing that we'll cover in this class, which is in some cases it is signaled that the cell is supposed to die now, as opposed to waiting for the default pathway to kick in. In that case, you have actual death receptors that are involved in that. So we'll talk about the so-called extrinsic pathway of apoptosis on the last day of class. But the bottom line is that we have all these complicated series of pathways that ultimately point to the nucleus and regulate genes, as you might guess. Relatively few things you'll notice from this diagram point out, and that's how we're going to wrap up the semester basically in talking about what those things are. And I can tell you it's going to be cyclins and apoptosis factors as the principal outcomes of all of these pathways. And it's the synthesis of all of that information in the nucleus that leads to what we hope to be the logical, coordinated pattern of cell growth and senescence and quiescence that is necessary for a multicellular organization to survive. And then we'll talk a little bit about what goes wrong when the process breaks down.